Stria terminalis, amygdala, and temporoparietal junction networks facilitate efficient emotion processing under expectations. Introduction. Rapid emotion processing is essential to survival. One needs to be able to differentiate between a friend or a foe. If a threat is expected, then one must be able to tune in to aggressive cues and appropriately respond. The same goes for when a non-threatening situation occurs. Thus, to process emotions quickly and correctly is vastly important. However, the underlying neural, functional, and structural circuitry is poorly understood. Previous studies have determined the amygdala's network as a core circuitry in processing emotions, but this study focuses on another neural network. This study seeks to investigate the neural networks underlying the fast recognition of happy and angry emotional expressions in conditions where these emotions were either congruent or incongruent with prior expectations. The researchers wished to relate the functional networks to structural networks previously linked to emotion processing, and they also wanted to include the relationship to emotion processing speed. The neural area the researchers focused on was the stria terminalis. Materials and methods. The study utilized 46 healthy right-handed males with a mean age of 39. The study utilized a dynamic emotion perception, a DEP task, where participants were showed various cues and frequency displays to elicit prior expectations during viewing of angry and happy audiovisuals. The DEP task was completed during fMRI. For the experiment itself, there were 48 emotional videos in total. There were 16 emotional videos for the conditions of anger, happiness, and neutral emotions. A female actor spoke 16 sentences in an emotionally ambiguous tone. This was to control the linguistic confounds. The cue was a still frame photo of the actress expressing an emotion, either happiness, anger, or neutrality, with the expressed emotion written in text underneath the picture. Then a video was played either congruent or incongruent with the previously depicted picture. A congruent condition is when the cue matched the video, and an incongruent condition was when the cue and video did not match. During the experiment, brain regions of interest were tracked via fMRI and an online SL Harvard-Oxford atlas. Data analysis was then determined via a multivariate partial least squares (PLS) approach for the functional/structural connectivity analysis and relating behavioral performance to the delineated networks. And this is a picture of the videos and the cues. So in black you have the picture where the actress was depicting in motion and then the motion was stated, and then the video was then played right after. The results. The analysis with right amygdala identified 2 statistically significant LVs. This suggests that the faster the recognition of happiness, the stronger the activity in this network. The functional connectivity in this network is then associated with greater FA in the inferior longitudinal fasciculus during anger congruent with cues. The faster the recognition of anger congruent with cue also depicted a stronger activation and microstructure of the right amygdala and hippocampus extending into the mammillary bodies, caudate and subgenual anterior cingulate cortex. The slower the recognition of anger congruent with cue depicted an activation of the left middle and medial frontal gyrus, bilateral inferior frontal gyri, left superior parietal lobule, and other closely associated regions. Conclusion. Ultimately, it was determined that the brain networks that helped recognize emotion changed depending on whether the participant expected to see the emotion with which they were presented. The amygdala network facilitated fast recognition of anger when people expected a threat, whereas the attention network was important for recognizing an unexpected threat. This research was ultimately conducted in hopes of discovering how those with psychotic and anxiety disorders may process emotions, because they have an altered stria terminalis pathway, and how this information can better help them with treatment. Additional research is hoped to be done with female participants, where their stria terminalis differs from the male stria terminalis. Thank you.